Good morning all. Today we can discuss the rest part of the aerobic culture methods. Today we are going to discuss a few methods coming under aerobic culture methods. First one is stab cultures. The stab cultures are similar to agar plates, but they are formed by solid agar in a test tube. The bacteria is introduced through an inoculation needle, or a pipette tip is stabbed into the center of the agar, and the bacteria grow in the punctured area. This stab culture method, it is the most commonly used for short-term storage or shipment of cultures. This is the picture of stab culture. It is performed by a straight wire charged with culture material by puncturing deep inside the agar. And while we are performing stab culture, we are not touching until the tip deep of the tube. Stab culture, we are puncturing the agar up to this point. Some of them are puncturing up to this point, but it is better to puncture up to this point or this point. There is no need of touching the deep end. And this is used to demonstrate gelatin liquefaction. That is a method. Oxygen requirement of bacteria, whether the oxygen is required or not, to check whether oxygen is required for that inoculated organism. To maintain the stock culture for the preservation of bacteria. So this is an excellent method for preserving the bacteria, to maintain the bacteria in the viable state. Next is about stalk culture. Stalk culture, it may provide a pure growth of bacteria for carrying out slide agglutination and other diagnostic tests. For this slide agglutination and this other diagnostic tests, stalk culture may provide a pure growth. And it is usually carried out in tubes, usually containing nutrient agar slopes. This is the picture of stalk culture, and this is the tube. Nutrient agar slope is made, and we are using this inoculating loop or needle. We are just drawing a zigzag pattern over the agar slope. And this stalk culture is made in tubes containing agar slant or slope, and this may provide pure culture for the slide agglutination and other diagnostic tests. Next is about lawn culture. The lawn culture provides a uniform layer of bacterial growth on a solid medium. And it is carried out how? By flooding the surface of solid media plate with a liquid culture or suspension of bacteria, pipetting of the excess inoculum, and finally incubating the plate overnight at 37 degrees Celsius. So simply we are flooding the surface of the media with liquid culture or suspension of bacteria. And we are excess inoculum is pipetted out. Alternatively, the culture plate may be inoculated by a sterile swab also, which is soaked in liquid bacterial culture or suspension and incubating overnight for the demonstration of bacterial colonies. So we can perform, instead of performing the flooding the surface of the solid media with liquid culture, we can take a swab which is sterile and dip it with the bacterial growth suspension and then incubate, or so not incubating, apply on the surface. And this can be done in the case of while we are performing antibiotic sensitivity test. So the spread or lawn culture plates It may result in a heavy, often confluent growth of culture spread evenly over the surface of the growth media. This means that they can be used to test the sensitivity of bacteria to many antimicrobial substances. For example, mouthwash, garlic, disinfectants, and antibiotics. This is the picture of lawn plate. So using a swab, we can dip on the surface, spread on the surface. So the lawn culture are obtained by flooding the surface of the plate with suspension of bacteria, mainly used for antibiotic sensitivity testing and preparation of bacterial antigens and vaccines, and also in the use of bacteriophage typing also we are using lawn culture. Next is about liquid culture. Liquid culture, it can be prepared in a liquid medium and it is enclosed in tubes, flasks or bottles. Liquid culture. The medium is inoculated by touching with a charged loop or by adding the inoculum with pipettes or syringes and incubating at 37 degrees Celsius, followed by, then after this incubation, we are taking that inoculum and subculturing onto solid media for the final identification. That is the method of liquid culture. The major disadvantage of this liquid culture is that it does not provide pure culture of bacteria, and also the bacterial growth does not exhibit special characteristic appearance. So we can't isolate a single or pure culture of bacteria using this method. And in the liquid culture we can't see any special characteristic appearance of the bacteria. So this is an example for liquid culture. How we are making the liquid culture. Next is the selection of temperature and environmental conditions. Once the medium is selected, temperature and environmental conditions must be considered. The environmental conditions are chosen according to the growth requirements of indigenous flora or pathogens suspected for the body site from the specimen which is taken. Fastidious organisms may require increased concentration of carbon dioxide, or they may require anaerobic environment for growth. Most routine bacterial cultures, they are incubated at 35 to 37 degrees Celsius for 48 hours. Broth cultures, when included, are routinely held 5 to 7 days. So the broth culture we are keeping for 5 to 7 days. Next is about culture collections. The microbial culture collections, it may focus on acquisition, authentication, production, preservation. So in order to, for example, when we are keeping this E. coli, for example, the culture collection of E. coli, and this may helps in the using that strains we can identify most of the characters. So we can preserve that E. coli and produce by subculturing, and we can say that that is the main strain of E. coli like that. So cataloging and distribution of viable cultures of standard reference microorganisms, cell lines and other materials for the research in microbial systematics, for all we are keeping the culture collections. So this culture collections consist of type strains. So they are representing all other strains. If for example, if the type strain of E. coli is like that, that E. coli is representing all other E. colis by showing the various biochemical reactions. This is the major nature culture collections. Then, so what do you mean by different types of culture collections which are brought? ATCC, American Type Culture Collection, that is located in Manassas and Virginia. So in our laboratory or hospitals, we are keeping the ATCC strains of various microorganisms. For example, E. coli, strep pneumo, staphylococcus, etc. We require an ATCC strain. Using this ATCC strain, we are subculturing that strain and using it as a control in many other biochemical tests, because it show all the tests of the typical E. For example, while we are taking E. coli and we are using ATCC E. coli strains, so we can keep that strain as a control to check out various biochemical reactions carried out by E. coli. And when we are performing a test, we usually keep a control whenever in the laboratory or in the hospital. So that control strains, we are using this ATCC or NTCC. So next is other culture collections are BCCM, CCUG, CECT, CIP, ICMP, JCM, NCTC. So ATCC and NCTC, National Collection of Type Cultures. So this both are used by ourselves. In most of our laboratories and hospitals, ATCC strains are commonly used. Next method is agar-based dipsticks. It is the miniature version of agar plates, which are implemented to dipstick formats. Example, dip slide, digital dipstick, showing the potential to be used at the point of care for diagnostic purpose. And they have advantage over the agar plate since they are cost effective and their operation does not require expertise or laboratory environment, which helps them to be used at point of care. So this is the latest technique of culture, agar-based dipsticks. So we have already mentioned that the primary isolation media for the unusual and fastidious organism, they require special necessities. For example, temperature requirement and length of incubation, it may vary with individual organism. We have told that fastidious organisms require 24 hours growth, and certain microorganisms, for example, tuberculosis, Mycobacterium tuberculosis causing tuberculosis, it requires minimum 7 days for the growth. Then unusual organisms may require special processing and selection of media beyond the routine. And example for such organisms are Mycobacterium tuberculosis. Next is solid plate cultures for thermophilic microorganisms. For the solid plate cultures of the thermophilic microorganisms such as Bacillus acidocaldarius, Bacillus stierothermophilus, Thermus aquaticus, Thermus thermophilus, etc. This Bacillus stierothermophilus we are using in the as an indicator for effective autoclaving and hot air oven. And they are growing at temperatures of 50 to 70 degrees Celsius. Low acyl clarified gelangum has been proven to be preferred gelling agent comparing to the agar for the counting or isolation or both of the above thermophilic bacteria. Next coming is virus and phage culture. Viruses or phage culture require host cells in which the viruses or phage multiply. For bacteriophages, cultures are grown by infecting bacterial cells, and the phage can then be isolated from the resulting plaques in a lawn of bacteria on a plate. And these are the phage cultures for the bacteriophages, that viruses which attack bacteria. The virus cultures are obtained from their appropriate eukaryotic host cells. So mostly these viruses are cultured in host. It's a picture of phage assay technique.